It's Hannah Johnson from the BI here, welcoming you to the one how flexible manufacturing can help your biopharmaceuticals. Welcome G on this webinar, so we'll be handing over to Paul Orange soon. Just a few things before we start. The webinar is about 30 minutes, and we're leaving all questions to the end. So if you've got any questions, either chat to me and I'll read them out, or we'll be unmuting everyone. A quick note before we start: we'll be muting everyone apart from everyone who's speaking, just to make sure there's not any background noise. So on no further note, I'll pass over to Paul.
Okay, thank you very much, Anna, and thanks to the BA for inviting us to talk today. So good morning, everybody. My name is Paul Orange, and as was me today on the call, I'm joined by a couple of colleagues, Pego and Madhu Raghunathan, who are going to help me with questions, and are also quite expert in bioprocessing. So I'm going to be moving through the slides pretty quickly to give you the opportunity to ask questions. We really are spanning the globe. Peggy is up late in Taiwan, and Madhu has got up very early in, I think, New York this morning. So thank you very much to both of them for joining us. If we look at why we're talking about biomanufacturing, and just sort of attention to that bottom line on this slide, manufacturing is something that enables biotechnology, and biotechnology has very exciting potential to really transform human health, saving even more millions of people, and hopefully doing so in a cost-effective manner. So we look where we are today with biotherapeutics, with strong presence of monoclonal antibodies. We're seeing vaccines make massive changes to human health. Certain diseases have been eradicated or all but eradicated from vaccines. You kind of fast forward into the future and you look at things like stem cell or regenerative therapies, which have the potential to not only cause us to be able to treat disease effectively, but potentially cure it with a single treatment. Very exciting elements of personal health and medicine that are enabled by biotechnology, but with manufacturing to support it, it's a very difficult prospect. There are things happening in the biotechnology market that we have to be aware of, and they inform some of the decisions and ways of working that we're looking at. So if you kind of think about pharmaceuticals in general, I think a lot of us know that we're seeing fewer blockbusters around these days. A lot of drive from governments to have in-market, for-market manufacturing, and those two together do give you small markets, maybe more niche markets. Certainly when you think about monoclonal antibodies, we see some of the original drugs that are starting to come off patent, the generic versions, biosimilars, and also better versions, so enhanced versions starting to come out. And at some point this is going to lead to a certain amount of pricing pressure. And surround this, this is a business for a lot of people, as a lot of companies need to be able to function profitably in this area. And the financial environment now is tighter than it used to be. Isn't so available. There's a lot less appetite for risk. That means you've got maybe sometimes less time in between different tranches of cash being available. And we've also seen a more aggressive partnering environment, and also merger acquisition and consolidation. And for those of us in the UK, there's been a lot of speculation around what's going to happen with GA at the moment as a good example of what's happening to companies within this space. Biotechnology products have been very successful. So if you look at the start of the century, you can see the significant growth in biotherapeutic products. And if you look at the red line, which is the total here, since 2001, this represents something in the order of a 15% compound annual growth rate, which is really quite phenomenal over sort of a 13-year period. And you also see monoclonal antibodies, which is the light blue line, is driving a lot of that growth. The compound growth there is around 24%, again, really quite significant. But what's interesting is, looking at all those different lines, there isn't a single one that's showing decline or leveling off. So definite growth around the world in the utilization in the market in from biotechnology-based products. We realize that we have to get better with these products. And I think what I like this particular description is, if you look down to the sort of the boxes at the bottom of the slide, you'll start off with a variable input. And I think for biopharmaceuticals, this is a lot more true than it would be for a small molecule compound, where chemical processes tend to be more understood and controllable. You start variable input because you're generally asking a cell to make a protein of some sort that you're then going to purify. Where we are today with current GMP manufacturing is we have a very fixed process. People spend a lot of time designing that, and then there's a lot of adherence to that process. So variable input through a fixed process. The end product, has to be consistent. It has to be safe and effective, so that you have some high defect rates. And to give you an idea of the defect rates, within the pharmaceutical industry overall, so this probably underestimates it a little bit for biotechnology products, you look at something like a 4 to 5% failure rate coming from the manufacturing process. And when you do that in terms of overall cost of goods, it's probably in the region to 25 to 30% of the overall cost of goods. So really significant impact on the production costs coming from your failure rate. We move to a more science-driven approach, which we've kind of talked about here as the better manufacturing processes. With adaptive processes through manufacturing, you understand a design space or a process space where it's amenable to be into. You can much more easily accommodate the variable inputs that you get, keep consistency of product, and get to lower defect rates. Aspirationally, an industry that's best in class here would probably be the semiconductor industry, where typically their failure rates are in the low part per billion, and their cost of defect here is probably like 1 to 2% of the cost of goods. So there's a way for us to go here. A lot of things that can enable us to get towards this state, so to run effective manufacturing processes. So typically has been a plant that looks something like the image on the left of this slide. Lots of stainless steel, very big, very capital intensive, quite often dedicated. Has now moved towards single-use technology, which enables quite a lot of the flexibility. But that on its own isn't enough. I'm going to talk a little bit now about some of the technological advances that have helped us get to single-use being more preferable, but it's not enough on its own. So I also want to look at some of the other things around single-use that really help you be successful in looking at making biopharmaceuticals. And I'm going to touch a little bit on how this gives you advances in 3 main areas: cost, quality, and speed of your product. Interesting slide which talks about a little bit about grounding us on what we need to produce. So if you think about the previous slide where you had those stainless steel plants, typically to produce large volumes of antibody. And these, I think, are the top 10 best-selling biopharmaceuticals in 2012, and they're ranked by revenue. You see all that are highlighted in blue here. So there's only 5 products that actually have production values annually over a ton of product, and you will see in the other 5 significantly less. And these products are, manufactured, exported, and used globally. With those drives away from sort of some of the big disease areas that these treat, and also for doing things on a smaller scale, what we want to see now is the need to not be producing at this large scale, but to be producing at smaller scale. It also has lots of benefits as you go through the process, which I just want to touch on for a couple of minutes, and especially as you go through the process of setting up a production plan, how some of the use of single-use technology or disposable technology actually allows you to be more effective with using your capital as you go through that process while you're scaling up, and particularly times before you move product when you're not seeing revenue coming in from it. But one of the technologies that's really changed that enables us here is looking at the level of antibody expression in the upstream part of bioprocessing. So this is essentially the cell culture part before you collect your crude product and go through the purification, sterilization process. So a major biopharmaceutical manufacturer. And what we talk about here is how the titer of the antibodies has changed over time. And you can see some of the products for big diseases like rheumatoid arthritis and cancer, their expression levels were down below 1 gram per liter. And as we've gone over time, we've reached 4 to 5 grams quite happily and even gone beyond that. And this is important because what I talk about it tells you how this can enable you to get better efficiency out of your plant. And again, just to give you some kind of a grounding, if you were to compare being able to produce antibodies at around that 4 to 5 gram per liter and produce it in a single-use environment, you're saving something like 60% of your variable costs compared to using that same antibody at a lower titer in a fixed stainless steel plant. So antibody titer is a great enabler of moving to these flexible products. And there's a sweet spot here for production. So the blue zone of the chart there, you see between 2 and 5 grams per liter production. And what you can see is if you're producing 10 or more batches per year, then your cost in terms of dollars per gram, at high end is about $400 per gram. And if you take it down to very high expression levels with high numbers of batches, you're down easily under 100 grams, dollars per gram. And if you start to go above 5 grams per liter in the titer, that can cause you some technical issues downstream. And in terms of what this has actually done in terms of antibody production costs over the past sort of 15 years since we turned the century is at the century, typically monoclonal antibodies to get you to your bulk final product, the production costs were somewhere in the region of $300 to $1,000 per gram. Really towards the high end of the chart that we're seeing here. Today, we estimate that that's probably closer to $50 to $300 per gram, and we're at the line of sight reasonably to getting down maybe to around $20 to $30 per gram in terms of production. So in terms of growth of the market has been great, but we've also seen massive efficiencies in the production, which, depending on how you cut it, are somewhere between 10 and 50x improvements in the cost of production. So to make these processes very efficient, enable you to work as well in these small scales as still in an efficient manner. And you produce less waste. So I mentioned a couple of slides ago about some of the earlier stages of bringing a drug to market before you've actually got revenue coming in. So typically thinking of setting up a stainless steel plant and you had something like a 10,000 liter reactor, you could see going on the chart here, which is the red line, really how effective your cell culture is at producing protein. You only probably need one run on that whole system to give you something like 10 kilos, which 10 kilos you're going to need to do that for manufacturing test runs, for some of your preclinical and clinical trial studies. But the investment in capital in setting up a big fixed plant at that level is significant. You're running to probably hundreds of millions of dollars and plus quite easily. If you can scale that down, so blue line shows you a instead of 10,000 liter reactor, a 1,000 liter reactor. You see that the sort of around 5 grams per liter in terms of product titer, it allows you to produce that 10 kilos in three runs with a much smaller capital investment. And also, if you have single-use technologies, those lines tend to be more flexible, so you can intersperse this with other things that those lines are doing because the chances of cross-contamination are pretty much removed using single-use. And this allows you also then to go from this flexible line, which is single-use, to scale up and to scale out with waste and less running costs as well. Antibody titer and the development of effective expression systems has really driven our ability to effectively produce now in these smaller scale, flexible, single-use production lines. Again, just want to reinforce the fact, especially at those points of time when you're not seeing revenue coming in because your drug isn't yet on the market. big factors that I wanted to cover: cost, quality, and speed. And I'll just move on to those and talk through them. So if you look at cost, we see interesting things around the process development, the system biology. With manufacturing, I've talked about single use gives you a lower capex expenditure. I'm going to talk a little bit as well in a couple of slides' time how it gives you shorter turnaround, more flexibility, so you can utilize your facilities better. But there's also, within manufacturing cycle, some interesting work done to make your cycle time as short as possible. So a couple of that was there was a recent publication which was done with authors working at both Genzyme and Wuxi AppTech, at shortening the upstream stage by doing perfusion cell culture at the N-1 stage. So this is the final stage of the seed train of your cell culture before you go into the production bioreactor. And showed, I think it was something like a 10% reduction in cycle time for that stage. But on top of that, some very clear ways how you could potentially get to a 25 to 30% reduction in cycle time. Cycle time reductions are really important when you've got a flexible facility where you have the potential to put multiple different runs through to produce those very high volumes, and we saw earlier that those very high volumes are not required that often. Then maybe a fixed plant that's able to run bigger batches is what you need, but cycle time then starts to again drive some real process and operational efficiencies within your manufacturing plant. I'd also just like to give you some examples of how you can utilize some of these process efficiencies. So here's an example, and this is our PCC system. So continuous chromatography is an example within that flexible approach. Helps you actually also to reduce some of the more expensive consumables that you're going to require during production, such as chromatography media and the buffers required for that. So lots of things happening to address cost, both from a technology and a utilization side, that flexible and single-use manufacturing is enabling. Quality is something that can never be overlooked, and it's the one thing that is, the absolute given that your product is safe and is developing the therapeutic outcomes for the patients who require it. And I think process development and manufacturing, an area that's really interesting to us, and this is an area that GE, an organization, is really interested in, is actually looking at data analysis as you're going through the process. So earlier slides where I talked about adaptive processes, one of that is absolutely the case is this significant trend within single-use and flexible to have more and more monitors and sensors within the manufacturing line, producing significant amounts of data. What we're then interested in doing is looking at that in, it's a big data problem, like the big data problem of for example, looking after an aircraft engine while it's up and running, and we kind of take some of those principles. So you look at that big data and you start to analyze the process. You start to understand where the efficiencies to be made and where there are changes that may need to be implemented. And an example I can give you on some pilot studies we've done is we've identified where customers are looking to replace their chromatography media because it's reached the number of cycles of regeneration that they would normally use. And we've actually been able to say through our big analysis, well, actually, you don't yet need to replace that because the media is still functioning perfectly fine within parameters. So again, that can, in that case, help you, lengthen the life of that media, which is a fairly expensive part of the process. On the flip side, we could turn around and say, that media maybe needs replacing slightly earlier, and that avoids the issue of having a failed batch, which has obviously a massive cost implication, potentially has an implication for your supply and patient treatment if you've got a very lean manufacturing and distribution process. So big data is I think, one of the very exciting areas, but also supported by very known quantities within the production process. An example here would be prepacked chromatography columns. We have a brand called Ready to Process. If you think about this adaptive process, having a very known entity in terms of how that particular element is going to perform, rather than potentially having a manually packed column or changing over in a column in a process. You start to then be able to be a little bit more intelligent in those big data analysis, and you're also able to have good sense of confidence about how that particular element is going to perform. And then speed, and speed to a certain extent is cost here. So there's absolutely a need, I think, for anybody looking to move into whether it's a biopharmaceutical or a small molecule compound, to move with speed. Whether you're using a generic, if you're quick, you're able to have an early mover into the market advantage. If you have a patented product, you clearly want to have as much marketing lifetime within the patent of that product as you possibly can, so that you don't suffer from competition from the generics. So there's lots of ways, once you've put that patent out there, that you can drive speed getting out to market quickly. So around process development, you can use tools like high throughput process development, where you're looking at multiple parameters, doing multivariate analysis. So rather than the more traditional approach, which had been optimize individual unit operations one after the other, you can optimize either segments or the whole process. And sometimes when you optimize the individual unit operations, you do get optimal output from step A, but you may find that then when you get to step C, step D, that output actually inhibits that working as best as you can. And what you're really interested in is optimal output when it comes to the bulk product that you're going to produce. And doing a multivariate approach really drives that. Also, on the manufacturing side, again, I'm going back to talking about full time, but there are things that you can do to skip hold steps, go to more continuous manufacturing, and I'll just mention that on the next slide. And then also the facilities that are easier to deploy and more off the shelf, and I will show an example of that as well in a second. Making your process easier, and this goes back to a publication, as you can see on the slide, from 2009. And moving between the different stages in your purification, taking it from that first polishing stage, putting it in a big hold tank, adjusting the buffer before you can then put it into the second polishing stage, and doing the same again. You can eliminate those steps by using a system that will allow you to concentrated buffer, dilute, inline, recondition, so you've got better utilization of your facility because you haven't got these big tanks that you've got to accommodate somewhere. You've got less chance of contamination because you haven't got product just sitting around in a tank and having people add these different buffers to change buffers around. And ultimately you get from your of the production through to a bulk product much quicker. If you've got a flexible facility, you've got multiple products going through it, your utilization goes up. So the technology is again driving some of these process economics very convincingly. Like an almost frivolous comment, but you have to think about how you construct the facilities. So you'd have something that looked like what you have on the left, a multiple clean room facility, dedicated parts of that facility, and it's kind of fixed. It's good for one purpose only, very difficult to reconfigure, and actually to get set up is fairly costly. If you have more of these flexible approaches where you maybe have the individual units as well are contained within sort of freestanding qualified container units, can connect in fashion to each other, you can reconfigure the space, and your setup for the building is actually much simpler. So you need, and I don't know how well this is showing on your slide, but you can areas where people for example, to gown up, you need some laboratories and warehousing, you need a sterile area to do preparation of your inoculum, but then the rest of it can happen in a non-classified space, but the individual units themselves are classified much simpler. And here, think about speed. It gives you a huge benefit in terms of your build-out speed, actually having your plant ready and up and running. And the ultimate there is looking for modular facilities that allow you to put everything together very quickly. I'll give you an example which is very recent for us, So we have this QBio, which is essentially a modular factory for production of monoclonals. Client out in China, and they're building a QBio. Now, typically think about a very linear, step-by-step process. You would start off, you look to, once you had a drug that you wanted to start going into some of your process development, taking through into your manufacturing, your preclinical, clinical trial runs. Very simply, you'd say, okay, I want to build a new line. I may need planning permission, so I apply for that. Once I have planning permission, I've probably got some architect's drawings, and I can get some builders on site. The build will take 12 months or so to build a factory. I then go through the validation, and then I actually start putting out my engineering runs. I can start putting out my preclinical test batches and my clinical test batches. For China, I got the planning permission for their site on Monday last week. They've actually been building their queue in our factory in Germany since January, and we started putting the module down on the ground Friday after they got their planning permission. And I was just before I jumped on this WebEx talking to our people out in China, and within a week we have the first floor put in place and the second floor starting to be assembled. Instantly starting to compress those timelines before you can get your plant validated and then start to do those production runs. So speed, whether it's that first mover biosimilar advantage, wanting to have as much of your patent protection lifetime as possible, we've enabled, somewhere between 12 to 18 months of additional time there, which is a huge benefit to anybody. And that you can do to accelerate that timeline, as well as just looking at the facilities. You can also, in parallel, do your process development. And some things that within that line, which in this are, as you can see, optimization in your upstream area, in your downstream. We can, scale up. One thing that's very interesting for anybody who's looking at biosimilars or maybe converting existing plants is converting from stainless steel to single use has some nuances to it. Again, we can sort of assist with those processes, and don't want to overlook the fact that we can help with a lot of the analytical services. Suddenly start to think out, well, I'm putting in parallel my planning permission for my site, very, very operational but very important. Production of the site is happening in a factory somewhere. And actually now what I don't want to do is then sort of say, okay, then have this big time lag of all this work in the new factory. What if I can do some of this elsewhere? So also to assist with that, we have something called bridge manufacturing, where up to 2,000 litre scale production, either for non-GMP requirements or for GMP manufacturing, up to and including phase 2 materials. So now you can see that you can get to the point, if you plan this thing very effectively, you can get having a factory that's qualified, you can have phase 2 studies completed, to start your phase 3 final production, final approvals in a short period of time, simply because you're putting these things in parallel. Great, but then you also need to think about who's going to actually operate the plant. And I think it's fair to say with biopharmaceutical manufacturing, there's a definite need for good expertise, which again, something that we can offer in many different elements, from actually putting our staff in the plant to help educate people who are going to be operating on the ground, GE training, qualified especially for you, or something that's more general for unit operations, documentation, general consulting and education. And when you start to pull all this together, what you get is interesting workflow of being able to take a provider that can, or a series of providers that can help you put your manufacturing processes in parallel. This is enabled by the technology, which is driving, for example, these flexible production lines, which are, I think, more suited to some of the market drivers that we see. As I said, things like niche products, in marketing, having in the market that you serve, having the training so you have the right people with the right education on site ready to run your plant, and to do deploy that plant at a very rapid speed compared to a lot of the traditional, more stainless steel approaches. So thank you very much for your attention. I am going to finish off there. I'll flash up the exciting disclaimer slide so that you're all disclaimed. But with Hannah, I think we're ready for questions. So both myself, Peggy, and Madhu would be happy to take any.
Everyone, so they should be able to talk. I haven't had anyone chat yet, but I'll open it up to everyone. Sorry, background noise. So if anyone has any questions, I have a few if no one else has one. Under what circumstances single-use technologies make most economic sense?
Well, I'll take a stab at that and ask Peggy to maybe chip in if they have any more insights. But there's one size of plan or design that fits all needs. Scale is important. The different markets being served is important. You have to think how frequently you're going to need to make the batches. Whether you're, depending on the stability of the product, for example. It also depends, I think, a lot on what your on-site capabilities are. There are some companies, the larger companies, who have a lot of experience in operating, steel fixed, and they may want to, continue operating in that manner because that fits their business model. So, there's those elements we really have to throw into the mix to end up thinking about what's the right use. And there are some things that you can do. So alternatives, things like intelligent column packing approaches. I haven't really mentioned continuous processing, which is a big drive within the industry. So moving from batch production to continuous production. Again, if you look at some best-in-class industries in terms of economics of their production, continuous production can be much more effective than working in batches. Things like some of the inline conditioning, so removing some of the hold steps, and again, that sort of processing, straight through without stopping, can be alternatives that make a lot of sense. I don't know if that answers the question, or Peggy, Madhu, do you have any other comments to make?
Madhu here. I think you captured the gist of that answer quite well. It will depend on the specific requirements in terms of scale, in terms of, often that particular batch is being manufactured. Is it manufactured for a clinical run? Is it being manufactured, for example, for commercial manufacturing, which means that product is being manufactured more frequently? That's going to depend on, like you said, the on-site capabilities that particular organization might have. So all of these factors come into play. So we have to look at all of these things in totality and decide what is the best approach. I can give you an example of one of the facilities that we recently went through in Singapore, where that particular organization actually ended up using 2-liter single-use bioreactors on the upstream side for cell culture. On the downstream side, they actually utilize columns, not single-use columns, but clean and reuse columns with intelligent packing. But what they did is they actually interconnected the columns in a see-through manner. Additional features like electronic batch records, online QC testing, and so on. So that approach actually worked really well for them, and also utilized single-use technologies on the downstream side for handling, for buffer storage, and so on. Long answer short, it absolutely depends on the specific production scenario and kind of the scenario plan for the future, and that's what's basically going to drive whether single-use technology, to what extent single-use technology is applicable.
Okay, thanks, Madhu.
some questions on the chat now. So
Yeah.
one from Sam from Faskins, which specific area is helping to make the most advances, chemistry, biology, or physics, and in relation to obviously the manufacturing advances?
Well, I don't know, if you're a reductionist, everything comes down to physics, right? So, I mean, I think at the moment one of the real big drivers is the biology around the tiny efficiency of cell culture. So, for example, host cells have improved, the vectors have improved, cell culture media has improved. Our understanding of some of the physics within the bioreactors themselves, so I think at the moment that's a really, really significant drive in terms of the efficiency and the effectiveness overall. So I mean, in that case, it's probably a mix of everything, right? I mean, cell culture media is a bit of chemistry, it's a bit of biology as well. So as somebody who's trained by a biologist, I'll claim biology is the bigger part of it, but I'm open for debate.
I know you touched on this earlier, but I'm just wondering if you've got anything else to add. It's what is your view on continuous processes?
Maybe I'd ask either Pam or Madhu to talk about continuous processes. They're probably a bit more expert.
Well, I'll talk about at least upstream continuous processing. I actually see a huge trend toward continuous processing. It does offer many advantages. There's an industry's trend certainly toward using continuous processing, as you mentioned before, for reducing the train. And today was interesting. I'm in Taiwan, as Paul mentioned. I have quite a few customers looking to do and link up continuous processing upstream, intensified perfusion to downstream processes to make just more streamlined and efficient, because much higher titers and product quality in some might be better because your cell viabilities will be higher. So may I have some comments about downstream?
Just to add on to what Peggy said about upstream, it's a similar mentality on the downstream side as well. If you start to look at, continuous processing makes sense. Like Peggy was saying, it's very suitable for unstable molecules and very tight product specifications. Then processing makes a lot of sense. Some of the other advantages that you also get is with smaller, you can actually go smaller in terms of footprint. Your processing time is going to go up. It also allows you to manufacture smaller batch sizes. It becomes more simpler. You're able to utilize your resin to a much better extent, for example. So there's a lot of advantages, and we see a growing trend in the industry towards adoption of continuous processing on the downstream side as well. Okay. Having said, what we are at this moment, what we are seeing is the greater adoption is in small scales of manufacturing rather than in the larger established commercial manufacturing, which are in the ton scale, for example. Right, that will probably come. It's more got to do with the fact that the traditional processes have been designed in a certain way, but what we're seeing is towards either fed batch or perfusion-based processing on the upstream side, and combining that with continuous processing and other process intensification activities on the downstream side as sort of a best class template on how you actually want to design your entire process.
Thank you. On the chat here from Steve. You mentioned High-titer. You hit downstream. What are they?
I think Madhu can talk about this, but a range of things from proteins clumping to, just over concentration. But Madhu specifically.
Only what I can actually do is have Peggy take the question first, because everything starts upstream, right? And if Peggy can take a crack at this, and I can follow up with the answer, which is probably kind of be a more logical response. Then Peggy, crack at this first, please.
Sure. I think most of the high-titer processes, at least in fed batch that I in the industry, more typically is in what Paul described as about 5 grams per liter. I would say from the upstream perspective, what customers have been beginning to see really around impact when you concentrate the antibodies in substance. So if you go to the literature, there's many companies that have started seeing that there is product quality, seeing that they have to modulate their cell culture media or remove these colors, like these in colors. So there's a customer, for instance, in substance, as they're getting to these higher titers and higher concentrations drug substance, they're going from pale yellow color, and they're starting to see a red color, for instance. They're doing is they're going in, and you can either remove your downstream, or you can say, I'm going to modulate and go look at my upstream cell culture media, perhaps. There's components in my cell culture media that can impact drug substance color. There's components like some trace elements, some B vitamins. So to run in at this point, modulate it, their cell culture media, optimize it with components to remove those colors that they can see, product quality colors that can be in the drug substance, and they don't have to do that in the downstream. But that's one of the things as you get to the high titer, some issues that customers have been seeing.
Yeah, so on from where Peggy left off, what we are typically seeing in terms of titers, established processes is probably around 2-3 grams per liter. And then start looking at newer processes, what we're seeing is titers going up to maybe 4-5 grams a liter. We expect that number to keep going, maybe 6, 7, even 10 grams a liter is possible, right? Like Paul mentioned earlier during his presentation, positive effects of high titers start to come off beyond a certain point. Now, to look at the upstream processes and downstream processes, the cycle time of downstream process is very different from the cycle time of an upstream process. Got to be able to balance that out in a way that makes sense for your entire process stream. Once you say that you have a high titer, and if you have your process properly on the downstream side, your result is not going to be significantly better, or it's not up to the required expectations that you had started going up in titer, right? So you've got to carefully design your entire process, your down matches up. The other thing also is when you go to the downstream process, and when for example, the concentration step, the ultrafiltration, diafiltration step, the concentration sometimes goes up to, what we've been seeing the recent past is starts up to 100 grams, 150 grams a liter. Is quite viscous, and that kind of poses some challenges, especially for example, if you're starting to use single-use, 2 bags or 3 bags, for example, for storing, and then for a recirculation bag when the concentration step, right? So you've got to carefully consider all of these different aspects in terms of how you size the process, what type of elements that you use when you're designing the process, and take into account the titers that you may have for that particular product that you're manufacturing or for other products that you will be manufacturing in the future. Look at everything holistically and make sure everything adds up. And, you know, that is a real important thing here. It's not just by increasing the titer, you can actually go up in productivity and everything else is going to work out.
Time for one more question. You mentioned big data earlier. Can you talk more on what GE is doing in this area to help biomanufacturing?
So, you know, one of the benefits of being part of the GE organization, which, you know, I'm sure you know, but, you know, GE makes everything from kind of light bulbs to locomotives to jet engines. We have a company-wide or accessible platform called Predix, a big software center out in San Ramon. You know, at the moment, the Predix platform is really an engine for analyzing big data. It's used. The example that I think most people kind of get their head around easiest is it's used on aircraft engines. So it's something. I mean, it's a ridiculous number of terabytes of data is generated during, you know, a typical transatlantic flight. And, you know, in the same way that they can be receiving information and they suddenly say, oh, number 3 engine on this flight is the oil pressure starting to look a bit high. They might want to start running it a bit lower, and we need to service it when it comes down on the ground. That's very useful because it optimizes and minimizes the downtime for aircraft and engines as much as possible for the industry. And this is, you know, something that really impacts their bottom line. So for, you know, you take that same approach and you look at the same data, you then kind of can use that big data analysis, that same Predix engine on what we see coming out of the sensors, the outputs, the inputs on a production line. Like I said, we've done some pilot studies, and a result of that, we've been able to identify, you know, potential, you know, extensions of a service interval for a particular part of the process, or even where we say, you know, there's something's looking wrong, and we think you might need to go back and change this. And I think I gave the example during the presentation where we were able to identify, you know, one of the things that's in your chromatography resins, you've only got a certain number of cycles that you can really use those resins for. So what we were able to do was identify that actually, you know, the whatever X number of cycles was that the customer typically used, this particular resin was still behaving acceptably, so they could extend it beyond that X without any impact on the end product. So it's really about the efficiency of your plant and making sure you don't have failed batches. And, you know, if you need a service person in because there is an issue with an instrument, you know, doing it in an intelligent way, scheduling it, you know, minimizing downtime, or, you know, staving off a potential issue that's up and coming. So those kind of things we're interested in looking at.
We're just going to have to move on. So any questions that I haven't answered, I can put Paul later and he can give you an answer. Or alternatively, if you've got any questions, please email me and I will send them off to Paul. Final thing before we close, just some upcoming BIA events that you're interested in. So we've got our big Bioscience Forum next month on October the 15th. We've got the Bioprocess Conference, which I imagine a lot of you might be interested in. That's the 25th and 26th of November, and it's happening in Cambridgeshire. Just thank you to Paul and Madhu and Peggy for joining us today. It was a really good talk. I thought we got some good questions as well. So, John, thank you very much. Thanks for joining us.
Yes, thank you, Anna, and thank everybody who turned up.